So we're at exercise E. In exercise E, we're going to be using the light ring as a source of light for the photosynthetic process. We're going to also be using these little chambers. The chambers are going to contain leaves that we're going to put in there. And we're going to be using the carbon dioxide probe. These are connected to the LabQuest. The LabQuest will record the information or the parts per million of carbon dioxide through the process. So in photosynthesis, as you know from the last experiment, uses carbon dioxide. So I would expect that as the photosynthetic rates go up, that the carbon dioxide rate amount should go down. So we're going to be using the leaves that have been kept in the dark. I'm going to load them into my chambers. I'll show you in the light chamber because it's easier to see. So I'm going to try to keep the leaves in here in the dark so other students can get those, but I want to take leaves that are healthy and are not broken and put them inside the chamber gently. Try not to squish too much. It's difficult to get them in there, but you're going to do 5, 6 leaves or so. And sometimes it's easier one at a time, but just trying to get this in there. So I'm going to get them down in the bottom there, and I will then put the top back on here so the leaves stay in the dark. And then I will bring it over here. If I'm doing the experiment in the dark, I'm using this covered chamber. The covered chamber will keep the light out of here. But regardless, I'm going to be using the light ring. What I will do is plug in the light ring. I will let this equilibrate for about 5 minutes, and then I will start measuring the parts per million here, right? So I'm going to do one group in the dark, so one set of 5 minutes. Then I will take the leaves out of here and put a new set of leaves in here, and I will do another set in the light. So I would be doing it like this. Again, I would equilibrate for 5 minutes and then let it run for 5 minutes. After that, I would be doing another set. The next set would be with the green filter. And then I would also turn on the light. So in other words, I would plug it in just like the last one, put the leaves in there, again, equilibrate for 5 minutes, and let it run for 5 minutes and record my information. Again, the last set is what I'm going to do is I'm going to take the probe and the LabQuest outside with my chamber. The chamber is going to be the one that is clear, does not have a filter on it. I'm going to put new leaves in there and go outside in the direct sunlight. That will be my highlight. So I have 4 treatments. I have, just to recap, I have the one in the dark, that is the covered one. I have the one in low light, that is just the light ring inside the lab. I have one with green light, and I have one that is in highlight that is outside. And you can compare the results.